Well, thank you very much. I'm completely overwhelmed to be here tonight to receive this award. I'm going to save most of my thank yous till the end, but I want to thank the Lemelson Foundation and the Eureka Fest and MIT, Lemelson-MIT staff that have worked so hard to put together such a fantastic couple of days. I've been completely inspired by the students here and the projects here, and it gives me so much hope for our future. And I was able to attend some of the Invent teams, I guess it was about a month ago, and I tried to, everywhere I went, I said, you have to see what these students are doing. You have to see these inventions that they're doing, that they're inventing machines that they can look underneath the ice and look to make sure there's no one has fallen in, or new ways of transporting dolphins to try to increase the lifespan of dolphins to get washed up on the shore, or there was in helping pedestrians cross the street, and it just goes on, and I was just completely amazed, and I was thinking, wow, I wish I was like that when I was their age, because I really wasn't. And I want to thank Ian for his very kind words, and for Evelyn, who's one of my dearest friends, and also the godmother of my oldest son. To hear that from someone that inspired you and who you look up to more than anyone else in the world is just so humbling. And your friendship has meant everything, and your mentorship. And I believe that where I am today is really my luck in getting to spend time with you. So I'm going to start by talking a little bit about my career and about my research. And I was told to talk about myself, which I'm a little bit uncomfortable with, but I'll try to rush that part fast and then talk about some science. I'm very, very humbled to be here today, and I appreciate you all coming out to also help me celebrate my birthday. Also, tomorrow is my husband and my anniversary. I think it's 13 years. He thinks it's 14 years. But he's probably right. But we eloped, and so this is kind of like the big party we never had. So thank you for helping us celebrate that as well. So, as was mentioned before, I've really spent my career thinking about how biology makes materials and borrowing ideas from biology to try to apply them to, as Evelyn said, systems that biology hasn't had the opportunity to work with yet. I think life has done a fantastic job. Here we are, where these organic, inorganic composite materials were self-assembling, self-correcting for the most part. But I think life could have done better if it had even more opportunity, and I'll tell you about some of that opportunity today. Let's see, I have this clicker. So I call this Giving Life to New Materials. I want to mention, I say Angela Belcher and her dream team, and I've been so fortunate to have the most fantastic graduate students and postdocs and undergraduates over my career here at MIT and also at University of Texas. I've had 46 graduate students and about 18 postdocs, and I've had 120 undergraduate research projects in my group since I've been here at MIT. And if you were in the auditorium 2 hours before I got here, you'd realize that there's so much happening behind the scenes. I had at least 8, maybe 10 students running around fixing slides, building demos, working all night long for days and days so I could be here to give this presentation to you to hear today. So this really is my dream team. I'm also very fortunate to work with so many fantastic collaborators here at MIT. And people say, why don't you work outside of MIT? Because all I have to do is cross my hall or walk to the next building and find people with absolutely fantastic ideas that'll say, sure, I'd love to work here. I'd love to help. How can I help? And it's just a great place to work. And so I wanted to start off talking about just engineering and invention. So I'm an engineer, and I can say right now this is the most fantastic time to be an engineer because there are so many opportunities that need our attention right now. And I've given you examples of invent teams and some of my mentors, But what I've learned in the couple of months leading up to this, in the couple of days of meeting the students here, and the collegiate students and the high school students and their mentors, is that the future looks very bright. Our future looks very, very bright, and it's because of your imagination and because of your passion and your desire to make the world a better place. And so I really want to celebrate you, our future scientists and engineers, and what you have to bring, because we live in very challenging times. But I really think that many of the global challenges in the world are opportunities for us as engineers. That we have the opportunity to make an impact in the world and in many of these different challenges. And so some of the most challenging problems are energy and water, food, healthcare, whether it's personalized healthcare, or the really incredible work that you saw yesterday with Maria and Rebecca in engineering for the underdeveloped world. But it's just a huge opportunity, and we get the opportunity every morning to wake up and think about how can we make a difference. And we can make a difference through our inventions. We can make a difference through our teaching of undergraduate students and mentoring of high school students and middle school students. But you're the future. You're our great hope, and I'm very, very excited to feel so confident about our future. So the other thing that you've heard many, many different times over the last couple of days in these talks is it's all about teamwork, and it's completely all about teamwork. In my group, I have mechanical engineers and materials scientists and engineers and biological engineers and computer science and engineering and mechanical, and just all of us working together, along with biologists and physicists and chemists, working together to see how our different viewpoints can come together to try to solve problems and to make a world a better place. This is my first group at University of Texas. When I walked in, I had no idea how to be a professor. These are the students that I learned how to be a professor and working with, and they're a really fantastic group of students. And some of my groups over the years at University of Texas. And this is my current group of students right here, many who are in the audience today, along with students from my 2109, my biological engineering junior level lab, where they built their own solar cells and tested their own solar cells, which was a very fun experience to be a part of, and some former students as well. And so every example that we've heard it's really about teamwork. It's really about ideas that are beyond your own ideas and how they can come together. So where did I start out? Well, one of those pictures that flashed by really quickly was me as a 3-year-old. And I knew I wanted to do something important. I knew I wanted to do something to make a difference. And it's true that I've always been interested in the origin of life. I've always been fascinated by about how molecules came together to form more complex molecules. How did DNA form? How did the proteins that interact with DNA form? And how did they all come together to form the cellular structures? And wow, we're this huge self-assembling structure. And if you think about it and you dial down and you think about all the enzymatic reactions that are happening in every single cell right now in your body and how they're transferring information around to each other, you think it's just amazing that we exist here right now. I've been fascinated by that pretty much my entire life. I did my undergraduate degree in creative studies, which was design your own major. And it was because I was interested. I only had 2 requirements, and I petitioned out of one, which I heard had never been done before. And it was economics. So I thought, why would I ever need economics? And since I founded 2 companies, it would have been a nice class to have. But basically, I designed my own major where I could put courses together any way that I was interested in. I wanted to take extra physics classes. I wanted to take extra chemistry classes and geology classes because I was working towards my idea of how all these things were synthesized together. I was very fortunate to be able to do that at UC Santa Barbara. And it was during my a junior level physics class that I met Professor Paul Hansma. Has anyone ever used atomic force microscopy here? or heard of atomic force microscopy? Yeah, some people. So Professor Paul Hansma commercialized the atomic force microscope. He took it from kind of a lab idea to being able to have one in the lab now. And I'll invite anyone who wants to come see one in my lab to come see it. And my students are going, wait, not all at the same time. But I was in his junior level design class, and I said, wow, you know, I really, really want to be a physicist. And he said, it was already a junior at the time and almost done. He goes, you know, maybe you should be a chemist. And so I didn't take it the wrong way, really. And he introduced me to Professor Galen Stuckey, who introduced me to Professor Dan Morris. And I got to work between them for my PhD. And Paul Hansma, the physicist, he was my co-advisor. And it was Dan who introduced me to Evelyn Hu, and I got to do my postdoc in electrical engineering with her, knowing nothing about electrical engineering. And I should also say that my undergraduate degree was in creative studies, and I decided to do my PhD in solid state inorganic chemistry, having never had a course before. And so Galen really supported my application. They were like, why don't you be a biochemist? You have a biochemistry background, but I wanted to learn something new. And usually you don't do that at the PhD level. And so I went and I took all my entrance exams, and I scored the lowest in the class. I mean, I really bombed the entrance exams because I'd never had any of the courses. But by the time I finished, I graduated first in my class. And it all was a really happy story. But so this is my last day in Santa Barbara. I was there from undergraduate through postdoc, and I had a lab that overlooked the ocean. I could see the whales and dolphins go by as I was working at my lab bench. And I had the most incredible mentors and great experience. And I was getting ready. I accepted my first position at University of Texas, and I bought a pickup truck. And I was walking across the street to get in my pickup truck and drive to take my first faculty position in Austin, Texas. And this is my last day with my mentors, and they're all pointing what direction I should go in my career. Evelyn's pointing this way, Dan's pointing this way, and Galen's pointing this way. And so I took that to heart, and I made a combination, and I went out and started my own lab at University of Texas. And I was asked to give a commencement speech a couple of years ago, and I titled it "Being an Expert, a Work in Progress, but a Really Great Ride." Because when you first walk in, you become a faculty member, and this is for Rana, who's starting in a couple of weeks, and Janet, my friend, who's starting faculty positions in a couple of weeks. You come in and you have this lab, and you're learning to train students, and you really don't know what you're doing yet. And but you learn along the way, and I called Evelyn several times, within the first week of being a professor. What do I do now? What do I do now? And she was able to advise me. And so that's kind of my story of how I got here. I'll tell a little bit more about the MIT part in a little while. But my inspiration, besides being those really incredible mentors that I worked with, has been nature, and it's been the Abalone show, which you've heard about, and trees, and just so many examples of how nature does such a fantastic job of making materials. And I say, I want to do that. I want to take those same low temperature, low pressure, non-toxic routes to making materials, and I want to use that as a manufacturing paradigm to not make trees, because trees are doing a fantastic job, to not make shells, but to make batteries and to make solar cells. And my students, I think, had passed around some batteries that are powered. They're virus-based batteries that are powering LEDs, and hopefully you got a chance to push on it. This is another example here. It's a little hard to see with the lights, but this is my decoy battery LED. This is one that actually went to the White House because when my students invented it, it was a record-breaking a battery at the time, and it was made at room temperature and pressure, basically mixing it up in the lab and making a battery. And I'll tell you a little bit about that today. I've lost my clicker. There it is. We got it. All right. All right. So one of the things that we do is we work on the nanoscale, and everyone here probably knows nano. Raise your hand. You know nano. Everyone loves nano. And maybe you love nano because of the cool nano iPods or the nano cars, or most of us use nanotechnology when we put on our sunscreens, or the Wii. The Wii is very big in my house and uses nanotechnology, or antibacterial socks that keep your feet from stinking. So nano is very, very big, and so people think, well, nano is great because it's small, but it's great for so many more reasons just because it's small. It's almost kind of magical. When you take a lot of atoms that are packed together and you start to whittle them away where it's just small collections of atoms together, they behave differently than they do in the bulk. They have different optical properties, electronic properties, magnetic properties, and to us that's completely fascinating because it can really change many things in the world, which I'll talk about, from healthcare to energy and to other kinds of applications. So small is very, very big. Okay, I need a virus-based remote. Okay, so so I'm an engineer. I want to solve problems. My group are engineers. We want to solve problems. Every day we say, how can we make a better place? What can we do to make an impact on the world? And we're interested in nanoscience, but we're interested in nanoscience at the interface of biology. And we think that by using biology to interface nanomaterials, you can have an impact in energy, healthcare, and environment. And I always say I'm not accused of focusing. Never been accused of focusing. But it really, in our case, is all about atoms and all about nanomaterials. Taking atoms and arranging them in particular ways that give you improved properties, and that's what we're very, very interested in. And by doing that, you can have an effect on solar cells and batteries, fuel, CO2 sequestration storage. You can make more earth-abundant materials, cleaner mining, water purification, therapeutics, and imaging. You say, wow, that's such a big diversity of materials. What if you just tackled one of those? And we say, well, from a material science perspective, from a biological engineering perspective, it's all about the atoms. So whether those atoms are arranged in such a way to make your solar cell better or to make your cancer diagnostic better, it's all the same to us. And so when we think about future technologies, we think about starting with the beginning of the Earth. But here's the inspiration. It's this calcium carbonate-based shell that Evelyn mentioned before. This shell is 98% by mass calcium carbonate, chalk, 2% by mass protein, yet it's 3,000 times tougher than its geologic counterpart. And you can come up and play with it, as we've promised not to break it later. But it's this very, very tough structure. And it's made in room temperatures, room pressures, in the ocean, and doesn't use toxic materials and doesn't add toxic materials back in the environment. You say, wow, great material. But to me, what's even more interesting in that, when you have a male and female abalone, they make baby abalone. They make baby abalone, and they pass on the genetic information that says, here's the genetic code to make a beautiful material. Here's the DNA sequence. Here's all you need to make an exquisite material. It's all in my DNA. And here's a diatom. Some of you that use natural toothpaste might have used these diatoms this morning. They're made out of silica. They're made out of glass. They're made at room temperature and room pressure. There's at least 10,000 different species of diatoms, and they differ from each other based on this overall shape. This is a scanning electron microscope image. And they can be microns to millimeters in size. And see, there's a glass structures made at room temperature. How many people have ever made glass at room temperature? Here, not very many people. OK. But they have the genetic code that says, here's how to make glass at room temperature. Oh, you want to have this beautiful structure? Here's what it looks like. No, we said, well, let's do the same things with solar cells and batteries. So going back to the idea, beginning Earth about 4.5 billion years ago. It only took about 1 billion years to have life on Earth, about over here. And then it started, and really incredible things started happening. You had unicellularity, you have multicellularity, you had organisms that could pass on genetic information, you had photosynthesis. You had the ability of organisms to take sunlight and make their own energy. But you know what? They weren't making hard materials. They could not make calcium carbonate. And you think, how could an organism have photosynthesis? How could it use sunlight to make its energy source, but not make calcium carbonate, not make a shell? Well, it was during this time, about 500 million years ago, the Cambrian geologic time period, that organisms started making hard materials. Anyone know why? Well, there was increased calcium in the environment, there was increased silicon in the environment, and increased iron in the environment, and it became toxic. And the organisms took their proteins, and they started grabbing those ions out of solution and building structures like shells. But this happened about 500 million years ago, but it took organisms about 50 million years to get good at it. And so when I want to do that too, but when I recruit graduate students, my graduate students and postdocs all around here, I said, okay, here's a 50 million year contract. You know, do you want to join my group? I've never had any takers. And so I've devoted my career to trying to figure out how to beat that 50 million years, how to try to convince an organism to make a solar cell, make a battery, make fuel, but do it on the order of a PhD student at MIT. And so this is one of my favorite biomaterials. This is my son Brazos. This was when he was little, standing next to one of his favorite biomaterials, the Triceratops. And anyone who has or knows small children, you know they're incredibly complex organisms. And you know, convincing them to put their shoes on or something can be very, very complicated. So when we think about convincing organisms to work with a new toolkit, we think about simple organisms like bacteria and viruses and yeast. And so what we do is we genetically engineer viruses and bacteria to self-assemble and grow batteries and solar cells and fuel and electronics, and hopefully those batteries that you saw that were passed around today. So going back to the nanoscale, so I said, I hope I convinced you that the nanoscale is special. And I hope I convinced you that by changing things at the nanoscale can have a big impact. But this is showing you some relative size of biomolecules in your cells right now and how small they are. This is DNA here. This is the antibodies. These are globular proteins and ribosomes, the molecular machinery that make your proteins. You look at those and you say, those are beautiful. Those are only a couple of nanometers in size. Let's use those to build our nanomaterials because they've already evolved over 4 billion years. They're probably pretty good at it. Let's use those kinds of structures. And so this is our toolkit. Let's use cells and polysaccharide sugars and DNA and antibodies and proteins and protein cages and viruses. Let's use that as our toolkit to design new electronic materials. All right. I'm not doing this very well. Okay, so here's some natural biomaterials. Here's the coccolithophorida. It's a unicellular algae. This is a seashell. If you break that seashell, which I ask you not to, and you look at it under a scanning electron microscope, you'll see it looks like a brick wall. Do you see that structure? Those are actually calcium carbonate tablets. That's chalk that's made like a brick wall. Well, there's two secrets to this calcium carbonate. There's two secrets to the chalk. One of them I hope you've got from now is nanoscale. These are made on the nanoscale. The second is that they have proteins that make this chalk, and those have had the chance to evolve over millions and millions of years. So evolution and nanoscale to make really great materials. This is a diatom. This is from my undergraduate research project. When I was an undergraduate, basically studying silica-based materials. And this is a magnetotectic bacteria that makes small single-domain magnets. All these are made at room temperature. All of these are coated at the genetic level using non-toxic materials. And so this is actually looking at that abalone shell, and if you fracture it, you see it's made out of proteins, and these proteins are the secret. They're the secret sauce. They are involved in sculpting those atoms, making them just the right placement to give them this really, really strong, tough, and beautiful structure. Many people may be wearing pearls tonight. Those pearls are nanomaterials. Those are biological materials. You're wearing chalk. You say, why did those pearls cost you? You're wearing chalk. But that chalk has evolved over millions of years to give it that really beautiful structure. And so this is an abalone, actually, and it secretes proteins to make this shell. And these are some, I used to work in the black pearl industry. I can tell you that story if you're interested in, to try to make more valuable black pearls. It's actually how we bought our first house, because it paid better than faculty position. And so I said calcium carbonate, iron, silicon, but what about the rest of the periodic table? There's so many things on there that are interesting to us as engineers. And so there's this part of the periodic table which gives you semiconductors that are electronics. There's solid-state lighting, like some of the solid-state lighting awards that we heard about here. There's all these transition metals here that could be really useful. And so our job is to give the periodic table give biology the rest of the periodic table to try to learn with it. Now, I was very fortunate when President Obama visited a couple of years ago that he visited my lab, joint with Professor Paula Hammond, one of my very best collaborators. And we showed him our work, and he's very nice and very funny. And I give these periodic tables out to the entering class every year at MIT. It says, welcome to MIT, now you're an element. I think most people have them. They were handed out. If not, I have more. And I wrote that because once I got to MIT, I found it was home for me. And I felt like I was really in my element. So I have them made for the incoming class. On the other side are the amino acids, which would be great to know, that are functional parts of proteins. And so I wanted to give this to, I've given out 5,000 of these around the world. I wanted to give them out to President Obama. And you think about, how do you bring that up with the president? Oh, and by the way, here's a periodic table. And so my husband and I talked about it the night before, and I was really, really nervous about it. And so I said, after he toured the lab, I said, sir, I want to give you this periodic table in case you're ever in bind and you need to calculate molecular weight. And so he said, thank you, I'll look at it periodically. And here he is. He later gave a lecture where he was holding the periodic table and said, people at MIT, they give out periodic tables. You know, what's this about? And so now you have your own presidential periodic table, so feel free to take that. But that's really our inspiration, is how to convince organisms to work at the rest of the periodic table so we can have other kinds of important functionalities. And before I said, biology, Abalone have evolved the ability to grab these ions out of solution. And basically, they have proteins which are made out of 20 different amino acids which have different chemical functionalities on them. In the case of the abalone, it's very negatively charged. Anyone know a negative charged amino acid in the audience? Aspartic acid, glutamic acid. And they can actually grab calcium out of solution. They grab calcium and then carbonate, calcium and then carbonate, and build this brick wall. And we said, well, that's pretty good, but we want to develop a protein that can interact with anything that we want it to interact with. How do we do that? And we decided to think about that. And so we borrowed an idea from the drug discovery industry called phage display. A phage a bacteriophage is a virus. It's a virus that infects bacteria. It's non-harmful to humans. This one is non-harmful to humans. It doesn't even make bacteria very sick. But from a material science point of view, it's this beautiful structure that's 880 nanometers in this direction, so slightly less than a micron, and 6 nanometers across, and it has single-stranded DNA. This single-stranded DNA codes for the proteins that make it up. Now, we can go in and use little molecular scissors called enzymes, and we can cut that DNA. And we can cut that DNA and paste in new pieces of DNA so that we can start engineering this virus to have new pieces. Now, the 50 million years is hard for us to get around, so we're going to do 1 billion experiments at a time. Okay, and that sounds like, well, my students are great. My students work really, really hard. But we don't have 1 billion students in my lab. We have 16 fantastic PhD students in my lab and many postdocs. But each one can do 1 billion experiments at a time. And they can have, in a small test tube, you can have, in a couple of drops, you can have 1 billion different viruses. And what we're going to do is say, let's take 1 billion viruses and throw them at a semiconductor and see if any of them like it. Most of them don't. You're saying, okay, she is really completely insane, but she just won the Lemelson 500. Okay. But going back to what my grant agencies, when you become a professor and you're trying to get money for your lab, you have to write and ask for money in the grants. And the people that reviewed my grants were not as nice as the people who reviewed this my nomination for this. They said, she is insane. That's what it came back as. So I had one that says, she is insane, and it was like in all capital letters. That was all it said. And the other one said, this is a pretty good idea, but there's no way that she can do it. She doesn't have the background. So I went home and I cried. My husband remembers it. The Army funded me anyway because they knew me. And they thought, well, maybe she's a little insane, but let's give her a shot. And that's the way that I started my lab, and we've been doing that ever since. So I fell in love with molecules. I thought I wanted to be a doctor because I thought it was important, but I fell in love with molecules. And I remember the day I fell in love with molecules. And this is a model of one particular molecule called the M13 bacteriophage. And this beautiful model was made over the last 5 days, 24 hours a day by Sam and Stevie and Matt in my group. I'm serious, 24 hours a day. I could call at 2 in the morning, they would be there. And I want to tell you about why this virus is special to us. So the virus has single-stranded DNA in it, and it makes the proteins that surround this DNA. And each of these strands here is a single alpha helix of a protein. And this is actually 1 million times big, 1 million times longer than the actual virus, and 10 million times wider. Is that right, guys? Yeah, than the actual virus. And so it's not to scale. But inside of it, watch this, high tech. Inside of it, it has single-stranded DNA. Okay? And each one of these colors represents a different gene. And this is the truth time, guys. Press the button, Angie. Okay. Each one of them represents a different gene. And we can go in and modify these genes, one gene at a time. And by modifying these genes, we can modify the corresponding protein. So press. Modification in this gene, modification in this protein. Modification in this gene, modification in this protein. Modification in this gene, modification in this protein. So this is an incredible molecular toolkit that allows us to go in, make modifications. I had nothing to do with it. They taught me how to use it. We can go in, make modifications in the DNA and modifications in the protein and use it as our building block and use it to test. And hopefully I'll go back to that in a second. Okay, so this is our toolkit. So now what we do is we take 1 billion of those modified viruses in that small vial, and we force them to interact with a semiconductor material, anything we want. Most of them don't bind. They get washed away. Some of them will bind because they have the right chemical affinity, those amino acids that are on the back of your periodic table. We remove them from the surface by changing their pH. Viruses can't replicate themselves. We have to infect them into a host. So we infect them into their bacterial host. The host acts as a factory and makes 1 million copies. Now we have millions of viruses that have some affinity for this material. Now we force them to interact again. We say, aha, now can you bind again? We're going to make it chemically harder for you. Most of them can't. They fall away. It's kind of our Darwinian process. We go through this many different times, the survival of fittest, looking for the one virus that can grow a battery, the one virus out of 1 billion that can grow a solar cell, the one out of 1 billion that can detect 1 millimeter ovarian cancer tumors. And then we can make lots and lots of copies of it. So here basically you throw them at a surface. Most of them aren't going to bind. A couple of them stick. You wash them. And now you want to elute them. You want to take these and keep them and put them in their own vial. And so you lower the pH. You disrupt the molecular interactions. You can capture them now. You infect them into their bacterial host. This nice bacteria makes 1 million copies for them of us and then spits them out. And now we have a purified virus that has a particular kind of application. It recognized that particular semiconductor. And so, And so what we're trying to do is give genetic information to non-living structures. And I was asked to speak at our president, Raphael Reif's celebration of him becoming president. And my students, the same students that made the phage, stayed up for days and made this cartoon. This is me, and this is my collaborator, Paula Hammond. And the dream is to give genetic information to solar cells, into batteries, into catalysts, into drop-in fuel replacements, into clean building supplies, taking waste CO2 from coal factories and making floor tiles that you can walk on, into detecting cancer in non-invasive ways, and using sunlight to make fuel and to make water. And the idea is the living, non-living interface, giving genetic information to these structures so that they can enjoy many of the properties that we have as living humans or organisms. And so this virus, as you see here, it's long and skinny. And what we can do is once we have these proteins engineered the way that we want them to be, we can have them grow materials for us. Not an abalone shell, but say we want to have them grow gold nanoparticles. Push the button. And so what they actually do is that now they can grab those ions out of solution and start nucleating and growing inorganic wires, just like the abalone learned how to grow inorganic materials. And so that's what we spent my career doing, and my students have worked on, is now using this as a template to grow inorganic nanowires. And so we can grow beautiful structures here. These are gold nanowires. We can give them the ability to grow gold and platinum in this particular case. And one of the things that we've been doing, so this is basically giving a gene to something to be able to grow an inorganic nanowire. But one of the things we really, really wanted to do was not give a virus one gene to grow a material, but give it 2 genes. Give it 2 genes to grow or assemble 2 different materials, because you can do so much more when you have that level of genetic control. So here's the idea. You have carbon nanotubes. People in the audience have heard of carbon nanotubes before. Excellent, guys. It's basically a graphene sheet, a molecular carbon that's rolled up to give really interesting optical electronic properties. But they're very, very small. Small is good. Small is bad. Small is hard to pick up. Small is hard to move around. But a virus can do it. So what we want to do was say, OK, we want to mix these materials, which are semiconductor materials, and we want to mix them together with these nanowires. And you can do it by physically mixing, but it's really hard to physically mix at the nanoscale. You can change the surface chemistry of them. But if you change the surface chemistry of them, it changes electronic properties. It changes optical properties. So you might have ruined what you were trying to do in the first place. But let's engineer a virus to be a mediator to one, grab onto a carbon nanotube with 1 gene, second gene, grow a semiconductor material, and build a new kind of hybrid material. Okay? You guys think that we can do it? Yeah. OK. So then the question was, how do you get from a DNA sequence to an actual device? These actual devices are things like these solar cells that I have in my hand that you can come up and look at afterwards that are virus-enhanced solar cells. There are these batteries that I showed before that are multiple genes controlling these batteries. There are these floor tiles that are made by proteins from yeast, actually, that took waste CO2 to build clean building supplies. So trying to take something that's wasteful and trying to make something useful out of it. So all of these are encoded at the genetic level. I knew I was going to lose it. Here it is. Okay. All right. So batteries. Here's my battery team. I have a fantastic battery team. Battery team, raise your hand. They're out here in the audience if you have a battery question. So basically, batteries, you have an anode, you have a cathode, you have a negative electrode material, you have a positive electrode material. In this case, are lithium-ion batteries, you want rechargeable batteries. The electrons need to be stored in one side or the other. So we thought, well, maybe biology can help here. Maybe we can use biology to make friendly electrodes. Maybe we can use biology to make better electrodes. And so that's what we've actually spent our time doing. So here, this was a former student of mine who's now faculty at Seoul National in Korea. His idea was, let's have a virus grow a cobalt oxide-based battery electrode, and then let's have it self-assemble into a battery. Aha! But if it doesn't work so well the first time, let's use evolution to make it better. Let's use protein design to make it better. And that's one of the things that I like most about this technology, is because if you can convince biology to do it just a little bit or at all, you have the possibility of evolution to make it better and better and better. So you don't want to throw out your first generation, because it can definitely become better. And basically, we grow them up in a flask in the lab, and then you have an electrode. You put down your cobalt oxide that's made by biology, a separator, and a counter electrode. So most of you haven't looked at battery electrode data before. Anyone in the audience? OK. Well, I saw a fuel cell, a biology. Yeah, there. I saw your battery. Yeah, your bacterial battery. And so what you know is that its capacity matters. How much, in this case, lithium that you can store and cycle. You want a rechargeable battery. This is commercially available batteries. This is our theory of where we thought the batteries could be. And we were able to hit theoretical capacity. But as you started to charge, it got worse and worse and worse and worse. So we were happy less happy, less happy, less happy. But still, when we maxed out here, we were still 2 times higher than commercially available batteries. Aha, and we had really smart students, and we had evolution to try to make it better. So my students went in, and they re-engineered it. They said, what we need is a battery that can grow a good ionic conductor for lithium, but a good electrical conductor to get the electrons in and out. So we're going to make a smarter virus. We're going to have one that can grow gold with one gene, and another gene that can grow cobalt oxide, and we're going to mix them together. And they did that, and they built really fantastic batteries. They also made them solution-based process, stampable, and worked on making them conformable. But then I went to my funding agency, and we said we made this great battery, we published it in the journal Science, and they said, oh, you're only halfway there. You brought me half a battery. Where's the other half? And so as I always do, I go back to my students, and I say, you gave me half a battery. Where's the other half? So they went back into the lab really, really hard, and these were 2 great students of mine, Yoongjung Lee and Hyungjung Yi, who both have gone on, are senior scientists and faculty now. And they said, we're going to engineer a virus to pick up a carbon nanotube. And we're going to engineer this virus to simultaneously grow a battery cathode material and then self-assemble into a battery. And I'm like, OK, well, OK, that sounds good. And so they made a battery that was actually an incredible battery. And that's the battery that I showed up here that we call the White House battery because it went to the White House. And it's a really long story that I can tell sometime. We then start looking at lithium oxygen batteries or lithium-air batteries. These are batteries that you're going to. This is the current batteries where you could drive with electric vehicles. And if lithium-air batteries take off, you're going to be able to go much, much farther. It's a huge hope for the future. It's very, very hopeful, but there's a lot of challenges to be overcome in order to do this. And we think that a virus can actually solve many of these problems. Or we think that biology and evolution can solve many of these problems. And so this is my student, Dayun, who's here somewhere, who's been working on this lithium-air battery technology. The idea was to get rid of some of the heavy parts of the battery. This wasn't her idea. This is the field's idea. And so that you're using oxygen as part of the active part of the battery. And so you can take oxygen from the environment and use it as part of the active part of the battery. And she's made a lot of progress on that, along with the rest of my team in the last year. Animation, keep going. Anyway, she solved all this. Maybe it has her dissertation in there. OK, and so basically, they've done a fantastic job of solving this through using multiple kinds of engineering, biological engineering, to look at some of these problems. Solar cells, an area that we're also really, really interested in. We're interested in inexpensive, flexible solar cells, and this is my solar cell team here. Most of them are here tonight. Some of them are on a plane on the way to a grant review for this program. They've worked on trying to make inexpensive solar cells. And these are called Dyesensitized solar cells. I thought I saw something on Dyesensitized solar cells here. Anyone work on Dyesensitized solar cells? Any of the students? So these are solar cells that are called artificial photosynthesis. They basically take light, and they excite a dye molecule. And that dye molecule takes the electron and injects it into titanium nanoparticles. And those titanium nanoparticles have to make all the way through these titanium nanoparticles to a current collector and then make the way back out again. And it's a really hard problem. Here it is. You shine light. The electron goes. It makes its way through. All these bad things can happen to this electron on the way through. They can get trapped by other molecules and never make it. They can get trapped by the dye themselves and never make it. So my student's idea, and Shengnan is one of the students that's in the audience tonight. My student's idea was to use a virus to solve this problem. Their idea was if you can take a virus and have it bind to a carbon nanotube, that the carbon nanotube could act as a pathway to directly deliver the electrons to the current collector, and that electron could be useful then. And they did this. They made virus-based batteries by taking 0.1% biology and adding it to a traditional dye-sensitized solar cell, and they increased the efficiency by 33%. 0.1% biology, 33% increase in efficiency. And they did that actually very, very rapidly. We've used these to actually do light harvesting experiments. These are viruses in here that bind to dye molecules to try to more efficiently harvest and capture light for many different kinds of reactions. It's a very, very fun experiment. Heachel's in the audience worked on this, and Nimrod is in the audience that worked on this, the virus-based nano antenna work. And this is a company I founded with Evelyn called Siluria Technologies. This uses the technology we developed in the group to combine it with rapid throughput screening to take advantage of one of our largest resources, methane, natural gas, to take methane and natural gas and convert it directly into gasoline and directly into chemical products. And I mentioned that, so we try to go directly from methane into useful things. There's at least 3,000 different products that you use right now that come from a molecule called ethylene. Normally, you get ethylene by taking large carbon chains and breaking them up into smaller chains. Does that sound very efficient? No. And so we said, what if you could take methane and start growing it up one carbon at a time? When you do that, you can reduce the carbon footprint by 70% in the synthesis of this material. And so people have been interested in this for about 30 years. A team at this company, Siluria, that I founded, solved this catalyst problem in about 18 months. Biology solved the problem in about 18 months, and it's being scaled up. We have 2 pilot plants right now making ethylene and California-grade gasoline, and as well as a pilot plant that's being built off the Gulf Coast right now. I mean, a demonstration plant. And it's all done by using this technology in rapid throughput screening. This is the CO2 building blocks that we've been able to make, take waste CO2, build tiles. And then we have a program in cancer imaging. And this is a program where my students developed a new way of looking deep inside. It's in animal models right now, deep inside the body to try to peer at very, very small tumors, tumors that are about 1 millimeter in size. And Rana is one of the inventors of that, and he's in the audience, and Neil is in the audience today. And they said, well, what if we could make something that was much cheaper than an MRI? What if we could do something in a very non-invasive way? And they developed a system where surgeons that we work with at MGH could find tumors that were less than 1 millimeter in size, which are really important for trying to beat some of the issues with ovarian cancer as well as prostate cancer and other kinds of cancers. And so I'm gonna run out of time. Can you guys go to the third to the last slide? This is what you're never supposed to do, especially in a big talk. There's all the things we're missing. Okay, there we go. So I want to thank Dorothy Lemelson for her vision in setting up a program that is so fantastic for students and future inventors. The Lemelson Foundation, Eureka Fest. I'm not going to thank all these people. These are the people that funded me. These are the departments that I'm in and the funding over the years, which have been fantastic. These are my collaborators and my really fantastic students, which I never say thank you enough. I say thank you yet again tonight. This is my husband and I, we eloped. This is our getting married on the beach in Hawaii on our 13th or 14th anniversary. This is me at age 3, standing next to my brother who's in the audience. And I was very lucky to be born to have a best friend. And so now that I have 2 boys of my own, I realize how nice it is to be born with a built-in best friend. And I definitely felt the same for me. And this is my 2 boys who mean everything to me. And what I want to say is, you're the pathway to the future. Don't walk, but run.